Okay, welcome to the third session of today's webinar, multiple sclerosis. Multiple sclerosis, in fact, is a chronic disease that occurs when the body's immune system behaves abnormally and attacks the brain, the spinal cord, the optic nerves of the central nervous system. And as we learned earlier, it's the leading cause of nervous system-related disability in young people. The biology is very complex. The cause of MS is not well characterized. Both genetics and environments appear to contribute to risk. More than 50 genes are known to increase the risk of developing MS, although none appears to have a large effect on its own, single effect on its own. Yet significant gains have been realized in the treatment of various forms of MS over the last couple of decades, leading to several classes of therapeutics, which broadly fall into immunomodulators, anti-cell trafficking, and cell depleting therapies. With more than 18 disease-modifying therapeutics in hand, the DMTs, many of which are mechanistically distinct, no single approach or single agent has produced sustainable relief for patients. Today we tackle and discuss these issues and look into the future with a distinguished panel of academic and industry leaders. And today we have Ralph Kern, the President and Chief Medical Officer at Brainstorm Soll, Lorrain, who's the CSO of Pipeline Therapeutics, Joachim Herz, who's at the Center for Translational Neurodegeneration Research at UT Southwestern, and Krish Ramanathan, the Global Program Head at Novartis. So each of you has had seminal findings, unique approaches, or recently approved products spanning the spectra of R&D space, from the discovery of targets to the approval of multiple modalities for therapeutic intervention. I would like to start with Krish. And Novartis received approval for drug multiple sclerosis in August 2020. Congratulations, by the way. It's always great to have a drug approval in the market for anyone. But let me ask you, what does the drug mean for patients? What does this drug mean for patients?
Thanks for your question, Rich. And thank you for the congratulations as well. It's a big sign of relief to the whole team working on this. So, at Novartis, we have a whole bunch of scientists here, and our job is we try to challenge medical paradigms and explore new possibilities, intervene in chronic illnesses earlier so that we can change the long-term quality of life. And we've been part of the neuroscience community for more than 75 years now. So coming back to multiple sclerosis, despite many treatment options in the current treatment landscape, there is still significant patient dissatisfaction. There remains an unmet need for early treatment with high efficacy therapies that can change the long-term course, and not a stepwise approach to escalate it into higher efficacies once patients begin progression. MS is a disease that's often triggered by an acute inflammatory attack. And we hope that with the drug, we're able to sort of shut down this focal inflammation that happens. It's a disease that starts off with the B-cell activation, leading to T-cell activation and eventual neurodegeneration. But if we can stop the B-cells from activating the T-cells, that's the hope with the drug. So on the MRI data, patients who went in with multiple lesions came out of the study with no lesions. So a 98+% reduction in lesion burden. So we hope that this brings a significant benefit. In the second year of treatment, if you look at it, patients who persisted with the treatment, 9 out of 10 treatments remained free of disease. And there's a term we use called NADA, or no evidence of disease activity. And you use either inflammation by MRI, or relapses, or progression. And so nearly 9 out of 10 patients remain free. So this brings hope, right? So, and finally, this is a drug that can be self-administered by patients. And that is a key criteria, especially in a pandemic. You don't want to spend hours sitting in an infusion center getting access to high efficacy therapies. So we believe that these attributes bring a lot of patient value in ensuring an early treatment with the high efficacy therapy that makes us a sort of a first choice treatment in patients with relapsing MS. So we have a portfolio of medicines. Each patient's MS journey is different. And so we hope that this gives the choice. But I fully attest to what you said in the beginning. There is still an unmet need in stopping the neurodegenerative aspects of multiple sclerosis.
What's the overarching strategy for Novartis in this space?
We hope that by earlier treatment options, we can stop the progression. So if you can identify those inflammatory triggers from the very beginning, because that's the portfolio of medicines we have medicines that can really interrupt the neuroinflammation process. So if you can identify either through biomarkers or through clinical history, identify patients very early in the disease, and if you can bring down the focal inflammation, it can lead to a significant amount of preservation of quality of life and reduce further damage that can come through neurodegeneration. We continue research activities in the neurodegeneration. We don't have a silver bullet yet, but we continue to do that. That may make complementary medicines to what we already have.
Thank you. Ralph, can you describe your platform, your technology platform? And at what stage do you think the therapy would be best implemented? Early stage, late stage?
Yeah, Richard, thanks for the intro and the kind words. We're taking a complementary approach to the approach that Krishnan has just described, and I was very proud of all of Novartis's accomplishments. I think they're really transformative for people with MS. I think the challenge really is that there still is a biological unmet need and there's a human unmet need, a patient unmet need. And well, really in terms of the neurodegenerative process, which begins immediately, it would be nice if it never happened, but I think that's the pipe dream. So I think the approach that we're taking is that ongoing compartmentalized inflammation in the brain and neurodegeneration, as well as loss of trophic support, in other words, the neurotrophic factor support, are all processes that are in play literally from the first day of all these diseases. And what we're trying to do is use a cell-delivered therapy. We're trying to deliver biologically relevant molecules that can address each of these three pathways by using people's own cells as a technology platform, creating a very pure fraction of these cells, taking the cells one step further to produce 5 to 10 times the amount of these repair molecules, and then delivering the cells directly back into the spinal fluid where they're free to travel and go to areas of damage and continually release their products. So unlike most biologics that are made in large bioreactors, we use nanoscale bioreactors. We deliver 100 million cells. The cells continue to produce their products, and these products impact those pathways. And the advances in medicine that have allowed this are, one, the manufacturing advances in terms of cell therapies. I think there's been essentially a paradigm shift in the last few years in terms of how well and efficiently the cells can be made. Secondly, the biomarker universe, all the way from in vitro through in vivo animal models and through to human biomarker experiments are essential, not important. They're essential to know exactly what cargo is being delivered, whether that cargo is being measurably increased in patients, and then whether that cargo delivery impacts pharmacodynamically the traditional disease pathways. It's different than a traditional molecule because you can measure PK/PD. All of my co-panelists are doing that quite assiduously. It's very difficult with cell therapies to do that. And in the CAR T space, they can't measure how many CAR T cells are there, but they can measure the impact that the CAR Ts have on disease biomarkers. We do a similar approach where we measure things like neurofilament, measure things like inflammatory biomarkers, measures of cell death, and we can show that we can move them in the right direction and then try to correlate that with the disease pathways that we know are important. So in terms of when is this approach ready for play, there's certain diseases like ALS where from day one people need it because there are no treatments right now that can impact a disease that has a 30-month average survival. For diseases like progressive MS, what we've done is we've selected patients who have entered the progressive phase, who have stable disease activity, and have a residual deficit that can't be addressed by existing therapies. And that's the area that we're looking at. In Alzheimer's disease, we've moved slightly differently, where we're using biomarker-selected patients who have specific disease biomarkers as inclusion criteria. And we think that this approach of for each disease, and I think for MS, there is a window of opportunity to intervene, and that's the approach that we're taking. And everything we do involves biomarkers. I think that's the way of the future, not just because we need it to confirm the cell's activity and their cargo delivery, but because we think that that will help us understand the clinical outcomes better. We think the regulatory world is moving in that direction as well. So I hope I've answered your question.
Oh, terrific. Thank you so much. Dan, first, congratulations on your financing last year for Series B financing, late December, I guess it was, in 2019. So my question to you is, how will this round be used to support the development of pipeline therapeutics emerging MS portfolio?
Thank you for that question. And as you well know, money goes fast. We're actually already knee deep into our next raise. But I guess you could say we're working on a more traditional approach. We're leveraging our chemistry expertise to design brain penetrant small molecule therapeutics to target remyelination. And, you know, I can say we all know and appreciate that MS is a chronic lifelong disease. And to date, as has been mentioned, there are several disease-modifying immune therapies that have been developed to treat MS, and they've had good success in controlling certain aspects of the disease. But unfortunately, as has been pointed out, at least currently, it appears patients, even those on the most potent immune modulators, still tend to progress, and that's a key issue. And we also know that disease progression or disability is inversely related to brain volume, and brain volume loss is a result of axonal loss, in other words, neurodegeneration. And this occurs following demyelination. So our goal here is to identify small molecule remyelination therapeutics with the goal of protecting axons, restoring function, and preventing disease progression. So our lead program is an M1 receptor antagonist. This is based on findings that came out of UCSF from our academic collaborators, Jonah Chan, Ari Green. I guess you could say this is a pure OPC differentiation play. So M1 is expressed in OPCs. Blocking the receptor, we know, facilitates OPC differentiation into mature myelin-producing oligodendrocytes. And we are in late preclinical development. We have a molecule that we call PIPE 307, and we're hopeful to begin early clinical development of this molecule late this year and into early next year. Behind M1, we have a heavy med chemistry effort right now on our second program, which is an LPA1 receptor antagonist. This receptor is also expressed on OPCs, and in addition to OPCs, it's actually expressed on mature oligodendrocytes as well. We know that blocking this receptor can also facilitate OPC differentiation and myelination, but there's a much broader play here in that we also know that LPA, the lipid ligand for the LPA1 receptor, is neuroinflammatory. So we believe that by blocking the LPA1 receptor, not only will we facilitate differentiation and remyelination, but we'll also be able to tamp down some of the neuroinflammation response that occurs in MS. And then behind that, we have a much earlier program with an undisclosed target for axonal protection. And so, our goal with the financing is to move all of these programs, each small molecule programs, into clinical development.
Thanks. So these targets are all mechanistically distinct, the programs that you're working on, and
correct.
It must be reflective of the complexity of disease, right? So then a couple of questions follow from that. What are the implications for at what stage of MS might they be used? And what have you learned so far from your preclinical studies, or even work that's been done in the clinical space with those mechanisms?
Our belief is that actually for all of these targets, we want early intervention. And really the next big thing for us is to figure out how we can combine some of these different targets, because we do think that they are complementary, and we need smart ways of combining those. We're working those out in our animal models now. And we also need to learn how to combine these properly with an immune modulator.
Great, thank you. And so Joachim, you had this very interesting Science Translational Medicine publication describing the role of the protein relin in multiple sclerosis. you're invoking potential use as a biomarker of disease and then also as a therapeutic target for therapeutic intervention. So what's the evidence you gathered on the significance of Relin with respect to either clinical correlates and also from your preclinical studies?
Yeah, so thank you first, Richard, for inviting me to serve here in this panel, and I'm really humbled to serve with that many of my colleagues here who know a lot more about this disease probably than I do. We came into this from the left field, studying Relin for over 20 years as a protein which is required for formation of the brain and for regulating synaptic homeostasis. And after we amassed various tools studying the function of this protein in the brain, we realized that it's not only made in the brain, but actually it's also made in the liver. So we were able to now test what it was doing in the periphery. This was initially really a side project, which we didn't really think would be leading to such a novel finding as the one which you just mentioned earlier, a role of this protein potentially as a therapy in MS. So what we did when we knocked out this protein in the periphery, in the liver, and tested its effect on the vasculature, what we found in a model of vascular lesion development was that Relin deficiency, so intervening with Relin expression in the plasma and neutralizing it, actually protected the vascular wall from lesion formation, specifically atherosclerosis. And when we followed the molecular mechanisms behind this protection, what we found was that Relin regulates the expression of endothelial adhesion molecules, which transiently trap circulating immune cells, attach them to the endothelial surface, and thereby induce them to listen for inflammatory signals from the subendothelial space, not only in the peripheral lesions, elsewhere in the body, but also actually in the brain. And by cutting this Relin signal off, there are fewer immune cells listening to these inflammatory signals, and therefore fewer chances for them to extravasate, leaving the vascular bed and contributing to the inflammation in the organ behind it. So we realized that this very general principle would be applicable to a very wide range of inflammatory diseases, among them MS. And since there are readily available animal models for multiple sclerosis called experimental autoimmune encephalitis, we could very quickly test this concept in mice, and that led us to this really very gratifying finding that intervening with circulating Relin in the plasma, which we can easily do by developing using monoclonal antibodies, which neutralize it, suck it basically out of the circulation, that this significantly curtailed the inflammatory process in the animal models. Now, you mentioned biomarkers, and the next step obviously was to see, well, this would also apply to humans. So in anonymized human plasma samples from control patients, MS patients with in relapse with an active disease, as well as MS patients in the remission, what we then found was that Relin levels did appear to correlate with the disease status. So Relin levels in plasma are higher in patients who actually suffer a relapse, and they go down again when the patient is in remission. And that made perfect sense for us, because Relin seems to be triggered, the expression of Relin seems to be triggered by inflammatory processes which are going on in the body. So if you're having an inflammatory process somewhere, those inflammatory signals seem to upregulate the expression of Relin, which is made mainly in the liver, actually peripherally, and circulates through the bloodstream, and thereby prepares the vascular bed for the eventuality that this inflammation might spread somewhere else by positioning immune cells at the site where they can very quickly then react. So increasing levels of renin in the plasma of a patient should be a sign of ongoing inflammation, and probably a sign that one should now intervene, cutting the inflammatory process. And one way of doing this is by using antibodies to suck out renin from the circulation, thereby dampening the immune system, reducing this exaggerated response, and thus curtailing the inflammatory process, which is so destructive on the brain.
So are you pursuing the therapeutic approach as an opportunity?
Yes, this is fantastic. As I said, this discovery came out, and this new concept, actually, what renin is doing, which we had no idea before we actually followed this seemingly wild lead. Why would we be interested in the protein that's made in the brain, but also secreted by the liver? When we found that this is such a general principle which is being regulated by renin with broad clinical application, this immediately told us that we had here an opportunity to move discovery out of the fundamental research arena and develop it towards the clinic. So this is something which I had never done before, and which I was very much looking forward to doing. So the way to do this, of course, is you have to move it out of an academic lab and do this in a dedicated biotechnology enterprise. So we founded this a new small company, which is really a fledgling company with the goal of developing anti-renin antibodies, which we can eventually put into the clinic. So this is now moved out of the discovery space into what I usually like to refer to as the engineering phase. And anybody who likes to tinker actually will be very excited in having such an opportunity in their life to follow. So I'm very much looking forward to this.
Thank you very much. So, it's impressive to see such a wide range of modalities being developed as potential therapies for MS. And we have, cell therapies, there's antibodies, there's small molecules. So let me start off with the small molecule team with Dan. Dan, and then I'd like to move to Krish. Maybe we talk about would different modalities be used at different times. So Dan.
Yeah, so good question. At this stage, there have been multiple targets identified, strictly speaking from a remyelination perspective. And, there's very good validation in animal models for a lot of these targets. But it's actually, no, it's not clear which one is going to work the best in patients. We're biased. Of course, we like our targets. From our perspective, we do have early indications of efficacy with a related molecule, clemastine. It's not an M1 selective antagonist, but it was a pan antagonist, and it did show some efficacy on improving remyelination in MS patients. But I guess multiple approaches, multiple targets, and ultimately these are going to have to be worked out in a clinical setting.
Hey, Krish, Novartis is taken on a number of therapy modalities in many different areas. How about in MS?
Yeah, I think it really depends on what is the target and what we want to achieve by it. I think we've early on we worked on small molecules like both the Gilenya and Mayzent are small molecules that stop the egress of T cells from the lymph and attacks into the brain. I think if you want to enter the CNS compartment, small molecules are the best way to do it. So you can permit the entry as long as they're not kicked out of the CNS. But if you want to act on the periphery, if you want to stop the immune cells from coming into the CNS and attacking, then the CD20 antibodies provide a nice way to do it, to interrupt the signaling so that at a more upstream level, so that you can stop all of those potential entry from the periphery. And also it gives a good kinetic profile because the B cell production takes some time, but with our therapy, which is an antibody that can be given as a monthly self-administration, you interrupt the cycle of production so you can keep those circulating B cells at bay and not coming from recurrence. And this stops the signaling cascade. If we were to look for neurodegeneration targets, I think small molecules make a good way to go in, because once the blood-brain barrier is closed, then it is difficult for these large molecules to otherwise enter and either start the remyelination or stop the neurodegeneration process. So those are very good for small molecules, potentially even gene therapies, if you can find a way to have a constituent expression in the CNS. So it really depends. We hope that potentially in the future there might be combination therapies. In other disease areas, like in rheumatology, people are comfortable doing combination therapy, but in multiple sclerosis and in neurology in particular, these are challenging based on, because you add the toxicities eventually. And even with just stopping these entry of inflammatory molecules into the CNS can sort of activate or permit the growth of brain-resident viruses like what we've seen in the past. So safety is paramount. We need to make sure that we can conquer that and still interrupt the process of neurodegeneration that can help progressive MS patients.
Would you like to comment on this?
Yeah, absolutely. I have a few comments to make. I mean, first of all, I appreciate Krishnan's comments. I think early in the, it depends on what the objectives are. I think that's really, and also the objectives change during the course of disease. So early on, there's certain objectives. Later on, there are others. Early on in MS, there seems to be a lot of peripheral immune activation, and I think that's where the targets are. And I'm hoping, and patients I'm sure are hoping that very early intervention with very effective treatments will nip it in the bud. The approach that we're taking is that later in the course of MS, there's compartmentalized inflammation, and it's less dependent on the blood-brain barrier and any other factors. It just takes up, the immune cells take up residence, and almost like local inflammation in other diseases have their own almost inflammatory organ that's operating. And what we're trying to do is we're trying to deliver the therapies directly into the central nervous system using the CSF pathways that are very well understood and they're used in other diseases as well. So again, and even within a group of patients, there are different objectives. So for example, addressing compartmentalized inflammation is one objective. Addressing the dysfunctional neuronal structures, loss of axons and synaptic loss is another objective. And then stabilizing the whole support system in the brain, this loss of trophic support is another objective. And essentially we're providing, multiple shots on goal, and it's impossible to know in any one patient which of these pathways is more important, because absent biomarkers that suggest which pathway is overactive in a given patient, you don't know if someone with MS might have predominantly a degenerative process or predominantly a compartmentalized inflammatory process. I think biomarkers will help tease that apart into patient subgroups, which will allow more targeted therapies. And then finally, the last thing I want to say is that we are doing combination therapies in MS. So for example, we allow disease-modifying therapies. We've done some very important interactive studies anticipating the high success of CD20 therapies, and we've ensured that the cells that we manufacture don't express CD20 on the surface, and they don't have gene expression of message for CD20 either. So I think that those are going to be the important preclinical studies that allow for rational combinations. First of all, making sure that the treatments aren't opposing each other. And then I agree with Krishnan that the next step is safety, and that, combining therapies might augment safety issues. And I think it's very important to do that. In our MS study, we're doing very careful monitoring of quantitative MRI to make sure that we don't have extra lesions. We want fewer lesions, not more. And we're being very careful. That's obviously something we're tracking very carefully. And then finally, the last thing is, even though there's biological processes that need to be targeted, one has to determine what's clinically meaningful for patients. In other words, what's the outcome that's valuable that will be meaningful to the patients and the society? And if it's improved function, then I think one needs to be very deliberate and precise about how one measures that. And that's one of the approaches that we're taking, is trying to determine, what are those objectives for functional improvement? In other words, what percentage improvement in walking speed or other levels of recognized MS function need to be targeted? And I think one needs to do all of those things well in order to advance the field. And I'm sure my colleagues are thinking about all the same issues that I am.
Yeah, would anyone else like to comment on that?
I guess maybe just one comment. Safety has come up a few times, and that actually is a key issue because with a lot of these therapies, they're going to be delivered for a lifetime, very likely. And so that sort of raises the bar from a safety perspective. And so whatever targets are identified or drugs developed toward those targets need to be safe with low side effect profiles so that they don't interfere with the patient's daily lives.
Okay.
And maybe one more point to add. I totally agree that safety is paramount here. Also, what Ralph mentioned on biomarkers or biosignatures, maybe multiple biomarkers that can sort of give you a peek into what is the phase of multiple sclerosis that the patient may be going through. Is it the inflammatory phase? Is it more progressive? And even within those, are there different flavors? We have a window into the brain through MRIs that gives you sort of snapshots and pictures, but they're not done on a quarterly basis. They're not done every time the patient's visiting the doctor. They're done every time there's a suspicion or a progression that is happening. So if there is something that we can query in the interim period that understands, is there neuronal loss, neurofilaments? I know there's tens and sometimes 100 presentations at these new conferences. I think we're also excited that this may give another peek or multiple peeks into the journey of the patient, because this is a protein that is integral to the cytoskeleton of neurons, and based on normal modeling of this protein, you see low levels in blood. And if you see a spike up, then there is certainly an injury in these neurons, and it must come from either an inflammation or a process that needs to be then considered to see how we can treat that. And it may give a window, it may precipitate an MRI. So these are things that I think certainly we can look forward to that may help the patients take charge of the journey.
What do you say is our best model in the case of MS for testing our agents before we go into human? Is there a particular model that it best reflects the disease?
So, I mean, we've been using the established experimental autoimmune encephalitis model, which is caused by immunization with an antigen which the body has, itself has. But clearly the etiology and also the immune targets in MS are broader than that. But the nature of the intervention generally preventing the accumulation of excessive more immune cells, recruitment of more immune cells from the circulation into the brain, where it then tends to exacerbate the local inflammation caused by microglia, should be applicable to actually any of the etiologies in MS. And I'd really like to hear what my other colleagues have to say to that as well.
So we use the MOG EAE model in our preclinical experiments, and basically delivered either no treatment, the undifferentiated MSC cells, or the differentiated product that produces higher repair molecules. And we compared the intrathecal administration of the two, of all three, either sham, undifferentiated or differentiated cells, and we saw, motor improvements and survival differences, the usual outcomes. I think the challenge with these experiments is that, I think the EAE model may be more useful for relapsing MS. I don't believe that there's a good model for progressive MS. I think these are necessary steps that everybody takes, but I don't think there's a good enough model yet for progressive MS. And maybe my colleagues know of one, but I'm not aware of a good model yet.
Maybe I could just comment.
Yeah.
So we also use the MOG EAE model, but we use a unique tool from a clinical translation perspective, and it's visual evoked potential. So we know VEP is used clinically in MS patients. We know when there's demyelination of the anterior visual pathway, VEP latencies increase. And that's because of the conduction velocity issues with demyelination. What's interesting, Ari Green, our collaborator, went back to the bench to figure out if you can use the VEP technique in an animal model And yes, he was able to show that in the MOG-EAE mouse model, there's demyelination of the anterior visual pathway that leads to a VEP latency delay. And with treatment, you can show an improvement in VEP, and you can correlate that to an improvement in myelin and in remyelination in particular.
Hey, is that used in the clinic, or can it be used in the clinical sense as a monitoring of disease progression, coming in early, for example?
Well, it is used clinically. It's been used in a few studies in MS patients who have chronic optic neuropathy or acute optic neuritis.
Okay, thank you.
I think that maybe to add to it, I think one of the challenges would be where is the location of the lesions? You know, if they have, as you rightly said, if they have chronic optic neuritis and there is a impact on visual evoked potentials, then it might give you an idea of the progression in these patients. Often the disease of MS itself is complex based on the location of these lesions. Yeah. That also makes it challenging to draw out, you know, meaningful conclusions from small animal models to then say what is the impact on clinical symptoms that you can have. Doing the EAE model, which is, I'm not an expert in preclinical science, but I've heard that is the model that we use in Novartis as well to screen some of these compounds. It's good from a broad brush description of the disease, but to look at, you know, minute symptoms of progression instead of just broad disability and just dysfunction, we need to really understand at a more complex level, and we have to do the human studies to understand how this can help with patient symptoms.
Sure. Okay, thank you. Ralph, we had a discussion around targeted delivery. So what do you see as, in particular, exosomes? Would you like to comment on that, please?
Yeah, so the cell mechanism of action that we're relying on is essentially a paracrine mechanism where the cells are releasing biologically active molecules and in the vicinity of other cells, and these molecules interact with receptors that are well known. The actual release of the biologically active molecules can occur through exosomal mechanisms or non-exosomal mechanisms. And what we've done is we've compared in preclinical experiments the amount of repair molecules that are released through each of these mechanisms, and we show that they're roughly comparable, and also the immunomodulation of cells versus exosomes is roughly comparable on a pound per pound basis. In other words, we have, let's say, 3,000 to 8,000 exosomes produced per cell, and we can compare the effects. So there are advantages of exosomal treatments. One is that it's easier to formulate, the stability. It doesn't involve all the other cellular machinery. If you want a pure targeted delivery mechanism, exosomes really do have some advantages. The other thing is that you can modify the cargo of exosomes. You can load, you can induce the cargo differently in exosomes, and you can also target the surface. And finally, you know, in our hands and others, we've shown that the exosomes are very avidly taken up by the target tissues, so that we can label the exosomes with various tracking molecules, and we can show that they're taken up into the target tissues, either in culture or in animals, and they can produce biologically relevant effects. So I think that's the next iteration of what we're doing right now. We've done some interesting experiments in different neurodegenerative diseases, in eye diseases, retinal disease, and also most recently we actually did some interesting experiments in ARDS because of all the interest in COVID. We're able to show that the exosomes, particularly from the differentiated cells, had profound effects on the target tissues, both in terms of decreasing tissue injury and also reducing inflammation, either cell infiltration or cytokines. So it's obviously a very active area of research for many companies. And down the road, it may be possible that one of the molecules that one of my colleagues is working on could be packaged in an exosome and delivered in some way, shape, or form. And finally, the last thing I'm going to say is that I still believe that local administration is the way to go right now, until such a time as the blood-brain barrier is sorted out, except for small molecules, which is a different kettle of fish. Very hard to deliver large molecules systemically. I think it's really not practical.
So what kind of technologies are we missing to advance some of these therapies into the clinic? I mean, we have technology gaps, clearly, we have to fill. Thoughts on what kind of key technologies you'd like to see or we need to move forward?
I think there are good technologies. We're not using them as well as we can. I think I think there are many studies that don't use enough biomarker assessments. I think that's a huge. We looked at clinical trial enrichment in different diseases, and such a small percentage use sophisticated methods. I think that's a missed opportunity. And then finally, I think there's a couple of approaches that might be useful. One is to combine clinical and biomarker characteristics as either inclusion criteria, or at least to stratify the results afterwards, to have pre-planned stratification based on certain biomarker categories. One has to start somewhere. I think obviously if you knew the answer, you would just do it. But I think that there's huge opportunities in many of the studies that my colleagues are doing to have pre-specified stratification based on certain categories. And I think that's the way to go.
Mm-hmm. Maybe I could just comment, bringing it back to remyelination. You can see where I'm focused. Yeah, so a key issue with remyelination therapeutics is imaging myelin and being able to detect remyelination in the human brain. It's easy to do in a mouse, right? You can do that histologically, not easy to do in human. The MRI techniques are getting better, but I think we need to develop myelin imaging tools. And so there's some nice work coming out of Case Western. We have some of our own work in-house that we're developing, but I think that's a key issue. That's one key issue, is being able to detect myelin, demyelination, and remyelination.
Okay. Anybody else?
Yeah, maybe just 2 points. One, I think certainly key gaps or key areas of improvement in technology for biomarker assessment. One is in imaging, as Dan, you mentioned, coming up with maybe more cost-effective imaging solutions that can give us either more frequent, looks into the brain, and also maybe on precision imaging on what's going on either in myelination or other areas at a more microscopic level. The other is blood-borne biomarkers. I think we have made a lot of steps because we are looking at a big dilution effect of anything that is in the CNS that's visible in serum. You're looking at picogram levels. You need to have very sensitive detection of these molecules of whether they're acute phase or markers of progression. I think we made some steps. I think we can detect some of these in serum today because we don't want to do intrathecal measurements of these biomarkers or CNS samples. So there are still steps, but I think how quickly they get used and incorporated into clinical practice, there's a few years off still.
Mm. What do you see as new directions in the area of multiple sclerosis? One topic that had come up in our discussion previously was that of antigen-specific therapies. Anyone would like to comment on that, for example, or other aspects of new directions going on in MS?
Well, one that comes to mind is absolutely, can you prevent the disease? Do we know enough about the disease to stop it from occurring? I mean, today the guidelines keep changing. We can detect it earlier and earlier from what constitutes multiple sclerosis. Is it a single relapse? You can look into the brain, you can look at blood markers and diagnose it earlier, but can you prevent it? Can you see who might develop multiple sclerosis, and can you develop immunomodulator or antigen-specific tolerization that might help prevent the first attack, that first B cells from turning pathogenic? It's still, our immune system is a thing of beauty. It doesn't break down all that often, but when it does break down, it goes into an irreversible mode. And we don't want the breakdown of tolerance inside the body. It would be great if we can find a way to tolerize patients to those. We're still far away, but this would be a big window into the future, into the next complete new generation of therapies or preventions.
Yeah, maybe I can add another area that I think would be, transformational would be to predict who's going to develop the neurodegenerative part of MS. Mm-hmm. It may be genetic tests, maybe other biomarkers, because look, there's a lot of people that have a lot of relapsing disease, but don't, for some reason, they have a resistance to entering that degenerative phase. And there's other patients who begin the degenerative phase right from day one. And there must be biological differences that explain that. And if one knew that a patient was at high risk of entering a degenerative phase, two things might happen. One is that a more aggressive treatment for relapsing remitting disease might be started early, because even a small amount of disease activity could push that person over the edge. Or there may be an introduction of treatment specifically targeting the degenerative component earlier on. And I think that type of precision medicine would be a huge advance. Obviously require a lot of scientific progress, but I think that would make a big difference.
Okay. We're just about out of time. What I'd like to do now is to just get some closing thoughts around the next generation of MS therapy. What are your views? What that next generation should look like? Maybe start with Ralph in this case.
Well, first of all, I hope all my colleagues are successful and they put us out of business. But, I still think there's going to be unmet needs because of the genetic heterogeneity in any human disease. One of the problems with the animal models is they're all genetically identical and all the humans are genetically different. And I think that until we can crack the code, we're going to have to come up with much more targeted therapies addressing different populations of patients, different stages of the disease, and also different components of the disease, looking at the peripheral versus central inflammation, looking at inflammation versus degeneration, and looking at the inadequate remyelination. So I think that that's a direction everybody needs to go in, and I'm hoping that targeted projects and programs that look at each of these parts will then be successful, and hopefully combinations of these will eventually be used.
Joachim, why don't I ask you to comment on, have some closing thoughts?
First of all, I'd like to echo that. And second of all, I'd say that our paper was published only about 2 weeks ago. And so we think that is actually the next generation, which we are going to explore for the moment and then see where that leads us. And we have very high hopes that this is going to be a very useful approach to support the existing therapies in combination with what has already proven to be fairly successful in MS, and hopefully we can contribute to with a new approach to lower the inflammatory threshold so much that the body actually has a chance to heal itself, and that may be one of my biggest hopes that we can achieve.
Dan?
I'm just excited for all the different approaches that are going on, and my fellow panelists and everything that you're doing. I guess my perspective is that from a near-term perspective, I think what we're looking at is combining something like a remyelination therapeutic with an immune modulator. And again, I seem biased, but one of the main reasons is that there's a crop of these remyelination therapeutics that are finally making their way into clinical development. And so I think we can properly test them at this stage.
Yeah, wonderful. And Krish?
Yeah, perfectly. I think complete agreement with all the panelists here. We have, I think there's quite a bit of new therapies to look forward to. With the availability of multiple disease-modifying therapies, we're able to tackle the inflammatory phase and give them a good course of the near-term disease. And I think with the level of biomarkers and other things that we can measure, also give a promise that they may have a better course in the future, maybe with new combination therapies and potentially even if we are successful with the neurodegenerative medicines that we have to stop the progression.
Well, thank you. I'd like to thank Dan and Krish and Joachim and Ralph for your participation on this drug development panel.